Thanks for inviting me to this symposium. So today I will focus on drug-drug preclinical interaction studies and spotting to some examples of Merck studies pointing to design and test article properties. So first, before to go to the topic, I would like to say I am from the original Merck, which is based in Germany, and there is the US Merck, which was based by a subsidiary of the German Merck, which was expropriated after First World War by the Americans. So, and now the US company is bigger than the original. So, the outline of my presentation will be to give you a very short introduction about preclinical drug-drug interaction studies. It can be a reminder for some of you, but due to I didn't know the audience, I will give a little introduction on this, and then I will go to 3 examples from studies that I have monitored in different projects for Merck. The first one is about impurity profile and influence on solubility of the test item. The second example will be relating to a CYP induction study showing the importance of the measuring and determining the exposure of a hepatocyte to the test compound by analysis of the cell culture medium during the induction experiment. The third example will be to an aldehyde oxidase inhibition study and about adequate selection of positive control inhibitors. So first, declaration of interest. I am acting here as a DMPK expert for small molecule at Merck, Germany, and I am involved in outsourcing drug-drug interaction studies at different CROs, including BioIVT, and more specifically Xenotech in Kansas City, and also acting as a project representative in several development projects or due diligence for Merck, mostly in oncology. Disclaimer, all the statements I will share today with you are my own and not representing the view of Merck. So, introduction. So, just a little reminder about drug-drug interaction studies. When you develop and during the development course of a drug to get market authorization by health authorities, the sponsor, the pharma company, has to complete a bunch of preclinical and sometimes clinical drug-drug interaction studies. These studies are dealing with the elimination mechanism of the drug by the body, which use metabolizing enzymes, CYP450, AOX, or other conjugation enzymes, or efflux transporter, which can be hepatic or renal. And the drug can act in 2 ways towards this elimination mechanism. So it can be a victim, and I heard today that in ICH 12 draft, they would like to use the word object. So if the drug is an object of a metabolizing enzyme or transporter, the sponsor has to characterize by which elimination route it occurs. And in this case, other drugs, which can be co-administered with your drug, can act as inhibitors of the elimination route and will lead to higher exposure and possible side effect of your drug. And this leads to notes in the labeling of the drug notice, in the package of the drug for the physician, regarding the adapting dosage eventually and the nature of the drugs to be co-administered. Second point, the second way is the drug candidate is a perpetrator or a precipitant now of the drug metabolism or transporter. So in this case, then the drug inhibits or induces your drug elimination route. So then the drug is now affecting the other co-administered drugs to the patient, which can lead to higher exposure if it's an inhibitor and possible side effect, or lower exposure in the case of an inducer, with the caveat that then eventually no therapeutic effect of the co-administered drug can be noticed. And again, this has to be put in the labeling drug notice. So I don't go to the health authority guidance document in extent to conduct preclinical studies. So you have I put in the bottom all the current regulations for your use, but some essential facts for this presentation are summarized. The first point is the sponsor has to show that the drug candidate used in inhibition studies, so in this case, in my presentation, in vitro studies, in the incubator is soluble in the incubation medium over the concentration range tested in the study. That will be related to the first example. In CYP induction studies, 1A1, 2B6, and 3A4, A5 are inducible enzymes. The sponsor has to show that hepatocytes have been exposed to the drug for the whole duration of the experiment. The experiment is usually 3 days with cell culture every 24 hours, by medium change every 24 hours. So to do this, the sponsor has to collect culture medium aliquots, which are sampled over the 24-hour period and analyzed to assess the concentration of the drug over the 24-hour period. Third fact is, if you pursue an inhibition study on one metabolizing enzyme or transporter for your drug candidate, you have to show the competency of the test system to assess this inhibition. This is done by using a validated inhibitor of the elimination route and showing that the inhibitor is behaving as expected, meaning relating to historical data that you have or to published data. This will be related to example 3. So first example, impurity profile of the test compound and influence on solubility. So I will relate it, to a substance from Merck. MSC stands for Merck Substance Compound, which was a tyrosine kinase inhibitor. So this compound, I cannot disclose the structure, is a lipophilic basic compound with a pH-dependent solubility. This compound shows tumor regression. So you see here a model of a gastrointestinal stromal implanted in the flank of mouse. These cells are growing, and you grow a tumor in a mouse, and then you can test your compound. In this case, it was used at 30 mg/kg once a day dosing. So then the control animals, you see the tumor growing. And ethically, you have to stop the experiment for the volume to the body weight of the mouse to a certain level, and you have to euthanize the mouse. And you see that this compound was very effective to do a tumor regression and even stabilization of the tumor. So the background of this case. So the solubility of a batch of the compound, batch 5, was granted at 25 millimolar. So I told you it's a lipophilic base, so it is more soluble at acidic pH, and so we chose to use pH 4 acetate buffer, and that was the limit, the solubility limit, 25 millimolar. Batch 5 has a purity of 96.8%, and there were no impurities which were higher than 0.5%. So I was the monitor of a study, which is a discovery CYP inhibition study over the 7 CYPs, which are the usual one dealing with drug metabolism. And it's a Xenotech special study which is called Discovery Plus. So during this study, the study was possible adding 0.2% final by volume of this acetate buffer pH 4 in the incubation buffer, meaning a maximum concentration testable of 50 micromolar. Turns out that the IC50 of this compound on CYP3A4 was reported around the limit of solubility, which was 50 micromolar. But then the compound was progressed towards IND submission, FDA, so we had to repeat the study to produce a report which can be used in the regulation, in the submission dossier. So the batch 5 was an early batch, no compound, so we had to choose another batch, A13. We decided to use this for the solubility testing and to repeat the study. So this is this. So, So what you have on the left is an extract of the certificate of analysis of batch A13. So you see that the purity is borderline. It's 95%, but it's fulfilling Merck acceptance criteria, which is 95%. But what was striking for this batch is that impurities have to be recorded if they are more than 0.5%. And there was a single impurity at 2.9%, which is quite high. But we decided to proceed with the study. So first, I told you you have to check the solubility, because then that will determine the maximum reachable concentration for your inhibition study. The solubility at the same concentration, 25 millimolar, in acetate buffer was done. But in this case, as compared to batch A5, it was not soluble. So here I put you an example of visual solubility testing. You have the buffer control, and here you have a compound which is not soluble. You have a A's. So I have a note. The case here is not the case of this one. It's a related case, but it's just to give you an impression to you of what solubility testing, visual solubility testing is. So in this case, the A's was less pronounced, but it was milky. The solubility was only granted at 10 millimolar, meaning that with the 0.2 final acetate buffer in the CYP inhibition assay, only 20 micromolar will be reached. So you remember it was 50, so that was not flying. So what we did then, and to move on, was we had no choice. We went to use an even later batch, A22, and this batch was purer, 88.3%, and cleaner compared to this one. with all impurities, again, less than 0.5%. On top of this, the solubility was granted, and to extend the maximum concentration testable, we checked with Xenotech that the CYP activities were not affected using not 0.2 but 1% of the acetate buffer in the incubation medium, and that was enabling us to test up to 125 micromolar. And then we were able to clearly have a good IC50 curve, enabling us to determine precisely the IC50 on CYP3A4, which was then 46 micromolar. By the way, this is an example of, usually you use, in the industry, you use a solvent to get stock solutions, and you then use a dilution, and due to the effect of organic solvent of enzymes, you use less than 0.5% of final organic solvent concentration in the assay. Here, it's an example which I think is interesting. This drug was very not good soluble except at acidic pH, and it shows you that you can use eventually an acid buffer to do your inhibition study, but you have to check that the CYP or the enzyme you are measuring is not affected. So the lessons learned are the following. First, you can ask the question, is a purity of 95% acceptable or not to run preclinical in vitro studies? Is a single impurity of 1% or higher acceptable? Is there any guidance of this? So from this example, I think I showed you that some impurities, in this case it was an impurity at 3%, can be less soluble than your test compound and polluting the visual test you do on solubility, which then will lead to an apparent lower solubility of the test compound, and you will not be able to test higher the concentration you want to reach to determine the IC50. So batch purity should be high, and as a guidance, it will be important to keep the impurities at low level. When you look at the guidance, then there is no statement about purity in the regulatory guidance to run preclinical studies, neither on impurity profile. Of course, it's different for the clinical batch you use in human. So with the example of this study, and from others, a good guidance will be to have a batch which is not a too early discovery batch, because usually the chemists, in my experience, produce impurity profiles and purity which are different from later batch. And also as a guidance, so remember you do preclinical studies, it will be good to try to use a batch which is close and consistent to the drug substance batches that will be used in clinic going forward, meaning matching the GMP, the manufacturing purity of your compound. So due to the fact now that I have 3 cases which are not related from each other, I would like you to ask questions if you have on this case.
Is it always 2 days? Is it 2 days or 3 days? Because I've seen some protocols for the incubation being 2 days rather than 3.
For the induction?
Yeah.
But I will go there in the second case. So here it was CYP inhibition studies, and CYP inhibition you do microsomes, usually human microsomes, and you test your drug as a potential inhibitor with all the substrates.
I have a question about So, it's impurity of around 3%. Is that structurally related to the compound to the point that it itself may have a pharmacological activity?
Yes, so I gave you the purity test is done by HPLC, so it's a kind of a crude peak area ratio. You are right, going further, when you go close to the clinical phase 1, we have at Merck what we call DP0, so pre-DP phase clinical 1. In that part of the development of a project, we can invest structural elucidation of impurities if they are prominent, and check the structure and the pharmacological activity of such compound. In this case, it was an early batch and we didn't invest in this. Yeah?
Yeah, so your solubility assessment was by visual inspection?
Yeah.
Do you think, have you measured actual concentrations of venom by, say, the LCMS? Do you think it would have had an impact?
So there was a lot of discussion about the high as an instrument versus doing a solubility test by, for example, spectroscopy. So we did that. I was on the edge to say that the high is very good, and apparently I was not far away, because in most of the compound when there was this comparison, the spectroscopy was matching the visual test. Do you have other questions? Or it's fine? I can move then to the second case. So it will be relating to a SIP induction study showing the importance to monitor the exposure of hepatocytes to the test compound by analysis of the cell culture media. So this case is relating to a SIP induction study for tepotinib. So tepotinib is now a marketed drug by Merck. Brand name is Tepmetko. It is a compound which is dealing with MET skipping exon 14 mutation inhibitor, meaning you have resistance mechanism for MET and they have mutations, and this compound is dealing with the resistance mutation of this MET gene. It's used to treat non-small cell lung cancer exhibiting this mutation, and you have a paper about that. So the background of that case is SIP induction studies by the drug candidate are normally, you have different setting, but the guidance say it's better to run this on fresh human hepatocytes, and they can be performed to look at two outcomes: SIP activity or SIP expression, SIP mRNA expression. Current regulatory guidance privilege the mRNA setting with a minimum incubation of 2 days, and they say it's up to the sponsor 2 or 3 days, but minimum 2. Outcome of the study is to measure and check induction of the inducible 1A2, 2B6, 3A4, A5. I don't go into the detail. If you have an induction of A4, A5, then you have to go then to see possible inductions of 2Cs. The threshold of positive induction of mRNA expression is twofold compared to the control, and you have to also have an induction which is higher or equal to 20% of a known inducer response. Meaning you have to use in parallel an inducer for the 1A2, 2B6 or 3A4, which the regulator recommends to measure this. In case of an induction, you have to describe the curve on induction with the Emax concentration and the EC50. And as I already mentioned, the regulator is asking that the sponsor will measure exposure of hepatocyte during the induction experiment to show that the hepatocytes were exposed for the sufficient time to the potential inducer. So this is the case. The study was done at Xenotech, and I was the monitor of this. So at the activity level, so meaning measuring CYP activity, there were no induction of 3A4, 2B6, 1A2. We did also the mRNA study for 3 days and changing and exposing the cells for 3 times 24 hours. It was in compliance with the EMA guideline. So tepotinib is also making a metabolite, and in this study we were looking at induction potential by tepotinib, but also by the metabolite. Cannot disclose the structure. We call it MSC1. The results of the study are in the table. So for tepotinib on 3A4, we use a sigmoid equation. I will go in a minute to that. So the EC50, we use 2 donors, or at least, in this table, 2 donors are reported, the 1249, 1252. And you see that the Emax was over 2-fold, 7-fold, and the maximal, the percent of the positive control response was not reaching 20%. So we were over 2-fold, but less than 20. So we are not, it's not a full yes for the induction response. For the metabolite, we had results showing that there was an induction with this donor for 2B6, and in this case, it was clearly over 20%. So it was a clear induction signal. For 3A4, same, it was 30%, 33% over the threshold. So then when you fit the induction curve, so it is suggested here in the graph, you can have different equations to fit the results. And there are papers outside in the literature trying to determine which would be the best method to fit the curve. Let's say that the sigmoid 3 parameters are leading to conservative estimates. When I say conservative, you can see that you can have a very wide range of Emax depending the equations, and the 3 parameters is giving you the conservative in the sense the lower Emax. So 3A4, I repeat myself, was shown for tepotinib, no induction to B6. For when you have an induction, you can then put the EC50 in in perspective of what is your clinical Cmax unbound value. And the cutoff value set in the guidance is that the C50 should be at least 50-fold the maximum, the Cmax unbound, which in this case was 2.6 micromolar. So we submitted the dossier, the market authorization submission, and health authorities question came back. So you have a summary of what we got about this SIP induction study. So the health authority was saying there was issues with stability of tepotinib and the metabolite during the induction experiment. The studied concentration were lower than the nominal concentration used in the study. So for tepotinib, studied concentration at the end were below the cutoff, 2.6. And then they said it cannot be excluded that induction of 2B6. Remember, I said in the previous slide that for tepotinib there were no induction of 2B6, but they say cannot be excluded that 2B6 induction could be present at clinically relevant concentration. So saying that we have to submit a new study or give a rationale for explaining the data. So the second part was more critical. The cutoff of the metabolite is 0.5 micromolar due to the limited, what the health authority is calling limited stability in the experiment, actual concentration substantially lower, which make the induction observed at 1, 3, and 5 potentially relevant. So discuss the reliability of this induction study finding, and you have to provide a rationale. So what are the, sorry for this busy slide, but you have the boxes. So for the tepotinib at the highest concentration 135, you see that as they say, we are above 2.6, but if you look at 3 and 1, we are significantly lower. By the way, you see that for each concentration, there is still the phenomenon that at the later time points, you really have a drop of the concentration compared to the time zero concentration. So for the metabolite, as they pointed out, so at 5 we are green because we are higher than 0.5. It was the cutoff for induction, but you see that at 3 and 1 we are borderline. So that was to explain what the health authority asked us to discuss. So the question then, how to defend the case and answer the health authority. So that was part of what we answered the health authority. So this is the first argument. So they say stability issues for tepotinib or the metabolite. So we answered that there were no issues relating to compound stability per se, meaning the stock or the dosing solutions at Cenotech were assessed and were fine, what we say fine, within 15% of the nominal concentration used in the SIP induction. On top of that, if you look at stability and eventually metabolic stability in the induction, the cleaned hepatocyte value was can be translated in an hepatic clearance in human, which was 0.27% of liver blood flow. So it's a very low clearance compound. So then the probability that the compound given in the SIP induction will be metabolized during the incubation in our sense, is low. So, but what was then true, instead of the wording stability, and this is from a recent publication of tepotinib by Merck, we showed that tepotinib has the potential to be trapped in lysosomes. So what you have here is a figure with two markers, a green fluorescent dye that can be trapped in lysosome, giving a green fluorescence, and also another marker which is a lysosomal protein. And you see that compared to the control, when you see the dye which is green in the microsomes, when you expose the cells to 10 micromolar tepotinib, you are able to blunt the lysosomal trapping of the green fluorescent molecule. So, if the compound can be trapped in lysosomes, yes, it can perhaps participate to the lower concentration that we were observing during the CYP induction experiment. So, then I come to the second argument we used to answer the health authorities. It was the following. Instead of just looking at the 24-hour last concentration, which was lower than the nominal, we ask ourselves, can the CYP induction be seen as a concentration mechanism, meaning Cmax, or more an AUC-driven mechanism? So then I am relating to a publication by this group in 2017, and these two figures are from this publication. They did, I think, a very nice experiment. So they were using an inducer of CYP3A4, rifampin, and they were able to, over the period of 24 hours, to give different schedules of rifampin. 1 hour per day, 2 hours per day, 4, 6, 8, 14 to 24 hours, the full 24-hour period. And then they used a concentration response from 1 to 24 micromolar on hepatocytes. And you can clearly see that depending the exposure, 1 to 24 hours, up to 6 micromolar of the inducer, you can see that you have really an AUC or a time exposure induction response profile, suggesting that yes, it seems that induction, mRNA induction of CYP3A4 by rifampin is more an AUC-driven mechanism than a concentration per se mechanism. So again, a busy slide, but basically you have seen this. So during the 24-hour, we measured at the different time points the tepotinib and here it's the metabolite. And what we did is to calculate an AUC based on the data I showed you, that CYP induction is probably more AUC-driven than concentration-driven. And we then calculate a C average, so we divided the AUC by 24 hours to get a C average over the 24-hour exposure of the metabolite. And in the last column, we normalized with the time zero concentration. So we had a C average normalized to the initial concentration, and you see that they are kind of remarkably consistent. So we took the average of all the CYP induction concentration we were using in this experiment. So saying to the authorities, we are now deriving a C average, because we consider the CYP induction as an AUC-driven mechanism, and not only looking at the last 24-hour concentration. So then what we did is to apply this C average normalized by C0 to the C50 of 2B6 for the metabolite, which was 1.9 micromolar. The corrected value was the multiplication of this, 0.97 micromolar, and using a free plasma metabolite concentration in human of 10 nanomolar, and applying a mechanistic static modeling. I don't enter the detail of what is mechanistic static modeling. The liver induction was 1.07. So remember, the threshold is 2. So using the rationale of an AUC-driven mechanism, we are able to suggest that there is no induction, 1.07. So we were then answering that this suggests, based on AUC-based average, that the induction on 2B6 by the metabolite was low. So, lessons learned. This example shows that culture medium analysis for the drug can be critical. So I showed you here that it was leading to questions to the market authorization of that compound. So we think that the AUC-based average coupled with mechanistic static modeling is probably a nice method to assess DDI risk for CYP induction. And here I have a suggestion for the design of CYP induction studies. Usually, and it was the case in that study, the culture medium analysis is done at the end, at day 3 of the induction. So it could be of interest, and I'm mentioning even for a clean compound. When I say clean, I showed you tepotinib was stable in the dosing solution. It was not metabolized, so we don't have foreseen risk that the compound disappear. But nevertheless, I showed you perhaps to binding to cells and lysosomal trapping, the concentration at 24 hours was lower than the nominal, the T0 concentration. So it could be good to run what I call a drug substance check on day 1, on the fly, on the CYP induction experiment, at the beginning of the CYP induction assay, to see if we will have a problem or not. And the second suggestion is to overcome possible decline. It could be interesting to replenish the medium during the 24-hour incubation with the drug substance during the day. So if you have questions, please ask them.
Maybe in the studies, given the requirements of the regulatory authorities that you're always above the threshold, basically this proof shows that you can overcome, even if you're below the threshold, that you can find a way to get around it. Do you think that the current rules by the authorities are too strict, or that they will be more contextualized to see average?
I didn't see. So we had this discussion with the authorities. At the end, they granted what we said. But if you look at I must admit, perhaps Brian can comment. I didn't go to all the comments of the ICH M12 draft. If there are discussions about the CYP induction design, I don't know that. But from my knowledge, no. Back to your question, no, they don't want to specifically go to this type of reasoning.
Though it's a nice
Yeah.
example of
Yeah.
how we can overcome it.
Mhm.
Looking at this, I'm thinking again of my own background with working on chipwood. What if those models become more and more popular? You can think of a system in which you have continuous flow of fresh media with fresh compound, which could potentially also be something that could overcome this issue for compounds with that stability. But that's still far, I think.
I think there were some comments that suggested using something like a C-average approach or an AUC. I think a couple of them came from Sotara, but as far as I can tell, those weren't included because in the YouTube video that was on from Kelly Reynolds and others at FDA and Health Canada, they mentioned that in some cases they were criticized for being too general and in some cases too specific. And so some people were asking for more specificity around this, and I don't think they're going to ask for more. Still just simply look at the split media analysis and then discuss it. They don't tell you what to do with it.
Also,
we have flexibility in some ways
Yeah.
to take this approach.
and also I didn't dig into that. So they are asking for a conservative mechanistic static modeling. In my experience, it's kind of it's a worst case. If you do physiology-based PK, you have usually the things are less worse that it appears by mechanistic static modeling, but they are still in the guidance pointing to mechanistic static modeling. And when using PBPK, then the sponsor has to provide a lot of data to show that the PBPK is behaving as it should. So I move to the next case, the last case. So aldehyde oxidase inhibition, adequate selection of the positive control inhibitor. So background, I told you in the introduction, so if you use an inhibition study for an elimination route, you have to show the competency of the test system. So this example is relating to an aldehyde oxidase inhibition which was run at Xenotech, and I was the monitor of this. So in that study, we decided to use phthalazine, which is a validated substrate for AOX and validated at Xenotech, as control substrate for AOX reaction. And we decided to use hydralazine, which is a validated inhibitor of AOX, but validated at Xenotech as control irreversible inhibitor of the aldehyde oxidase enzyme. So just a very short introduction. AOX is a very interesting enzyme. It's a molybdenum cofactor containing enzyme, also a flavin, flavin containing cofactor, and on top of molybdenum there is 2 iron sulfur center, so very complex from a biochemical standpoint. So just a short reminder. So you have AOX substrates which are relating to physiological molecules. For example, the retinal aldehyde, which is oxidized to acid, pyridoxal to pyridoxic acid. So here in a plant, vanillin to vanillic acid. So in the field of drug and xenobiotic, most of the xenobiotic can be aldehyde oxidase substrates if you have a carbon which is alpha to in an aromatic cycle to a nitrogen. So these positions are good substrate for AOX reaction. And it is the case from this phthalazine, which is regularly used as a substrate, which gives then oxidation by AOX to phthalazinol. and hydralazine can be an irreversible inhibitor of this. So, moving to the study, we used phthalazine as substrate, hydralazine as inhibitor. We used S9 liver fraction, so the cytosolic S9 fraction, and due to the fact that hydralazine is an irreversible inhibitor, so the S9 fraction was pre-incubated 30 minutes to give rise of the irreversible mechanism of hydralazine, and then followed by 10-minute incubation with phthalazine. So that was the result of the outcome of the experiment. So you see that there were no IC50 determined. There were scattered results, and even a tendency to lower inhibition at higher inhibitor concentrations. So please remember that we used S9. So in S9 you have AOX, but you have also CYPs there, which are of course less concentrated than in microsomes, but there are CYPs there. So busy slide. So I went through the literature to try to understand the results that I showed you, and this is the complete picture of what could be the hydralazine metabolism in human. So the major route is this one. The major route is acetylation. What you have in the big box here, which is very important for the case I showed you, is that hydralazine was used as an inhibitor, but it can be oxidized by CYPs, and remember we are pre-incubating 30 minutes with S9, and it can be also non-enzymatically oxidized to phthalazine, and further from phthalazine non-enzymatically to phthalazinone. So remember the substrate was phthalazine and oxidation phthalazinone. And even more, you can have spontaneous hydrazone here, and which can then degrade spontaneously again to phthalazinone. So you have multiple routes that hydralazine can form phthalazine or phthalazinone, which can then interfere with the reaction. So this is summarized here. So I have shown you on the previous slide from the literature that hydralazine can form the oxidized phthalazine product, or even the substrate product, leading to this, and which is, I think, and meaning either by the CYPs present in the S9 or spontaneously. So what we did is to use another validated substrate at Xenotech. So this is what is written. So we have to use a known substrate of aldehyde oxidase as a positive molecule to show the competency, and we used validated inhibitors, raloxifene, a direct inhibitor, and hydralazine metabolic dependent, irreversible inhibitor. And here you see that we were able to have a nice IC50 using paravanillin and hydralazine as reversible inhibitor. So now I move to what I said this morning. It turns out that there was a very recent paper by Pfizer team about screening of a library at Pfizer to try to find out a specific aldehyde oxidase inhibitors, but also other drug metabolism enzyme inhibitor. So AOX is not a specific inhibitor, hydralazine is not a specific inhibitor aldehyde oxidase. It inhibits CYPs. So this is down the paper. It also inhibits MAO and NAT2. I also showed you that hydralazine is not stable and give rise to phthalazine and phthalazinone. So the Pfizer team looked at more specific AOX inhibitors, and they looked also at other inhibitors for all these series of the enzymes here. So they screened roughly 5,000 compounds in their library, and they did it with a wise way. They had a cocktail of 20 enzyme substrate in a pool, and trying to identify specific hits, which were defined as 80% inhibition and less than 20% on the other drug metabolism enzyme. And the summary here for the AOX story is that they were able to identify two selective aldehyde oxidase inhibitor, erlotinib and dibenzothiophane. And in a follow-up paper, which is in press, they either were able to identify another one, an analog of erlotinib, as a specific AOX inhibitor. So lesson learned. So you have to carefully select the inhibitor you want to use to determine the competency of your inhibition route. Selectivity is very important. Here, remember, hydralazine is also metabolized by CYP to have a good potency and a stability in the incubation medium. I showed you that hydralazine was oxidized and giving rise to other compounds. Also checking that the inhibitor is not a substrate of the DME reaction it inhibits. It was the case here. So this field is evolving, meaning what to use, and the Pfizer paper, two papers, show that you can eventually, by high throughput or medium throughput screening, finding more specific inhibitor for AOX, but for also other drug metabolism enzyme. And as I said this morning, in that paper, they showed a specific inhibitor for all the CYPs, 2J2 included, CES, MAO, NAT1, NAT2. So, and at final, I want to acknowledge a member of my group, Ulrike, the head of the in vitro group, the boss, Dominique, the support for outsourcing study, Melanie and Ulf, procurement at Merck, the scientists which were included, study director in these studies, Deep, Julie and Jennifer, and the scientific management all over these years, Lois and Rebecca, and for many years as well for outsourcing support, the great support of Darina and Joanna, the head of Xenotech in Kansas. Thank you.